Howdy, my name is Robert Milliken, and the title of my report is How the Neglected Principle of Biology May Be the Driver of Speciation. Now, I attended Dr. Jeffrey Hill's presentation, which he titled Mitonuclear Coevolution as the Genesis of Speciation. Now, some background knowledge about him. He teaches and conducts research at Auburn University, and he primarily focuses on birds. Now, his presentation was very interesting. It immediately captivated me through just the title, as prior to his presentation, I'd never heard of mitonuclear coevolution. So I immediately wanted to learn more just about that. But I'll give a more detailed reaction to the entire presentation at the end of this report. Now, the question that he wanted to answer was whether mitonuclear coevolution did play a part in speciation. And if it did play a part, how big of a part did it play? Was it very significant or was it more minute and could be ignored? But before we can get into that, we should probably define what mitonuclear coevolution is. Now, as he defines it, mitonuclear coevolution is the process through mitonuclear coadaptations where nuclear and mitochondrial genomes adapt with each other and form to be one efficient block, basically. And if it's not 100% efficient, life could not be possible for the organism. It needs to be 100% or it just won't survive. It's very important. Like, for example, if my mitonuclear coadaptation was not 100%, I would not be sitting here right now. So before getting into his actual experiment, we should probably also cover how speciation is defined today. Now, allopatric speciation, which is commonly used, it only focuses on the nuclear genomes. So basically, you have a parent population, it breaks off for whatever reason, and it creates two daughter populations. And over time, they adapt with their environment through genome means, and they become two separate species. And then if they were to try to mate, it could not work. But he wants to also focus on how, whenever this is occurring, how do the mitonuclear aspects also change? And he does this through mitonuclear speciation. And it makes things a lot more specific and it also a lot more rapid because allopatric speciation could take hundreds of years. It could take a very, very long time. But this could occur within one simple population and it could just break off into two species just like that. Now, how did he test this whole idea of mitonuclear evolution and speciation? Through birds. Well, he technically was not the one that did it. He had somebody in Australia do it for him because he couldn't get it to work. Thought that was worth mentioning. And what he wanted to see was whether these long-tailed finches could actually be their own species. Because right now, under allopatric speciation, they're in a much larger group, right? But he believes that they could actually be their very own species through mitonuclear incompatibilities. So, how did this experiment occur? Well, he used the embryos of bred finches within the laboratory, and he tested to see their mitonuclear um, genomes. And he wanted to see if there was asymmetry within it, because if there was asymmetry within the mitonuclear genome, it would not live. He had to do it on the embryos because if they were to have been born, they would just die and he couldn't test them. And what he found was that within a lot of them, there were issues. There was asymmetry. And this meant that even though the finches look similar and through allopatric speciation, they were their own species, they could not mate with each other. And that's why the embryos would never hatch. And if they did hatch, they die immediately due to respiratory issues. Because as we defined earlier, if it's not perfect, they're not going to be born. So, in conclusion, this shows that mitonuclear coevolution is a very important way to define species because clearly it does play a very, very large role. And it's also very important because it could happen very, very rapidly. So the list of species that we have now is not accurate. So some implications with this is that right now we have to start applying this in order to have better, more serious, more specific species. And this could be important for various ways through not just having a prettier picture, but also with like global warming, as even though this population might be defined right now as one species, they might be two species and could have their own environmental needs, and we need to adapt to that. And my overall reaction was that it was a very, very good um, presentation. I enjoyed the little jokes that he applied throughout, and I liked his very simple slides. It made it easy to follow for somebody that had never really seen it before. And it was also very, very captivating and intriguing because, like right now, or last semester, I was in Biology 111 and we learned about speciation, but this aspect of it was never covered. So I would like to see if it would ever be added to the curriculum. So thank you.